logical place. I may even stop at some other place, but it will be likely the end of the lecture today. Yes. Next question is, are you doing a review session, Kevin? Does somebody want to ask that question?
Are you doing your review session, Kevin?
Thank you for asking. Yes, I will do one tomorrow night after class. I'm hoping to get this room again. I haven't confirmed that yet. And if it's not in this room, then we'll go somewhere else. And yes, I will videotape it and all that sort of stuff that goes with it and try to get it posted as quickly as I can. How's that? All right, so we're essentially done with glycolysis. I just wanted to say a brief word, excuse me, a brief word about this thing I talked about before, which was alcoholic fermentation and lactic acid fermentation, and just refresh your memories on that. So I showed this slide last time that showed in bacteria that and yeast that when they have run out of oxygen, they make NAD, which is what they're doing right here, by converting pyruvate to acetaldehyde. That loses the carbon dioxide. And then acetaldehyde to ethanol, which generates NAD, which can keep glycolysis going. Okay? This reaction that goes right here in bacteria is very similar to one that we run in our bodies. In our bodies, we take from pyruvate to acetyl-CoA, and that's what enables the citric acid cycle. And that process, which I'll talk about later, actually involves a coenzyme called TPP, thiamine pyrophosphate. It's one of the reasons we need thiamine in our diet. And again, I'll say more about that later. Okay. That is the reaction that's there. As I said, this is a little bit of an unusual reaction in that this is a decarboxylation that's happening in yeast, but it's not happening with an oxidation. So there's no oxidation that's coupled to this, which is why we don't see any NADs or NADHs popping up. It's a rather unusual decarboxylation. Most decarboxylations are coupled to an oxidation reaction, and this one is not. Okay? The enzyme that catalyzes that pyruvate decarboxylase, again, I will talk about when we talk about the citric acid cycle. And here's thiamine pyrophosphate. There's thiamine, vitamin B1, what it looks like. And that's what it looks like when you put two phosphates on it. This guy has a very important role actually in our bodies, in that instead of letting go of that acetaldehyde, which is what happens in bacteria and yeast, this coenzyme holds onto it, and that's why it never gets released as acetaldehyde. So as a consequence, we take acetaldehyde then and oxidize it to acetyl-CoA. So that's a two-step process that occurs in us. Both of them are bang, bang, right on top of each other. In yeast, they have an opportunity to let go of that acetaldehyde, and that's why they then are able to reduce it and make ethanol and make NAD. Okay, blah, blah. Instead, in our bodies, of course, what we're doing is we're taking pyruvate when we need NAD. And, of course, when do we need NAD? Well, we need NAD anytime we're short on oxygen. We'll see why this is the case later. But anytime we're short on oxygen, we have low levels of NAD. Very, very important point. So, for example, if I'm running faster than my blood can deliver oxygen to my muscles that need it, my muscles will be short of oxygen. And keeping those muscle cells supplied with NAD is necessary when there's low levels of oxygen, and that's when this guy kicks in. That's when we're doing fermentation. Yeast and bacteria are doing fermentation when we close the lid on them, of course, and they have no oxygen with which to continue their oxidation reactions, and then they start making ethanol as a consequence. So we're both doing it for the same reasons. We just make different molecules. In us, we're making lactic acid. In bacteria and yeast, they're making ethanol. Okay, you might wonder about plants. I didn't talk about plants in that scheme. Okay, nobody ever asked me about plants. All right, and the answer is that plants don't have common conditions like that where they're running out of oxygen because they're in an environment that is pretty constant. Okay, they don't have that need to have rapidly changing oxygen concentrations, and as a consequence, they don't really have to worry about this. Okay. I want to turn our attention now from talking about the breakdown of glucose to the synthesis of glucose and the storage of glucose. So this topic I'll be talking about today and on tomorrow will be called carbohydrate storage. And is a very important consideration. Now, what I want to do is talk about, not starting with glycogen, I'm actually going to start with gluconeogenesis right here because it's relevant to what we've been talking about. And then I'll come back and talk about glycogen metabolism. Okay? So gluconeogenesis, the name, again, tells us what it does. Gluco referring to glucose, neo referring to new, and genesis referring to making. So we're making new glucose. That's what gluconeogenesis is all about. Now you might think, well, why do we have to make glucose if we have glucose in our diet? And the answer is that we're not eating constantly. Some of us are eating almost constantly, but we're not eating constantly. So our blood glucose levels are changing. And if we go for those periods of time where, wow, I'm really starving, I didn't eat lunch today, or I'm really starving, it's 2 o'clock, and boy, I can't imagine it's 3 more hours until dinner time, or 4 more hours, or whatever time we eat dinner, then the body has to make up that. The body looks at glucose as a poison, but the body also looks at glucose as necessary. So the body has a very sort of dance that it has to do with glucose. It needs to keep enough in the bloodstream that our cells can have energy, but it doesn't want to get too much in our bloodstream or it'll kill our cells. So there's a yin and a yang to glucose. Well, there's 2 ways that our body has for maintaining glucose levels in our bloodstream. They both are done in our liver. Okay? One is the synthesis of glucose, which is gluconeogenesis. The other is the breakdown of glycogen. Glycogen, you remember, is the storage carbohydrate that is what has hundreds or thousands of glucoses contained in it. So by using these 2 processes efficiently and effectively, the body can release glucose as it's needed and maintain those blood glucose levels. Very, very important point. Okay. Well, gluconeogenesis you're going to like. And the reason you're going to like it is because it's mostly the reverse of glycolysis. There's only 4 steps that differ. Okay? I see some smirks like 4? Geez, I thought you said it was a reversal, right? All right. Well, let's talk about those 4 steps. It turns out that 4 steps in gluconeogenesis are necessary to replace 3 steps in glycolysis. That means one of the steps takes 2 steps to overcome, and that's the one I'm just getting ready to talk about right now. When we start talking about making glucose from scratch, which is what we're doing, okay, we start talking about making glucose from scratch. In this case, scratch is pyruvate. All right? We have to basically start at the bottom of glycolysis and move our way up. That's what we're doing here. Pyruvate, you recall, was the last molecule that was made in glycolysis. So it's appropriate that we start there and move upwards. Well, if you recall about the synthesis of pyruvate, I said it was a very interesting reaction. In fact, I described that reaction as the Big Bang. This guy was a reaction that was full of energy. Okay? And what we will see in the 3 steps in glycolysis that have to get reversed, all of them involve large amounts of energy. All of them do. Cells have to overcome these large energy barriers in order to make a reaction go forwards. When I talked about the aldolase reaction, you may recall, I said the cell did some tricks to get around the aldolase reaction. You may remember what they were? Pushing and pulling, right? So pushing and pulling was how the cell got around the aldolase high energy barrier. That's not an option for pyruvate because the barrier is even larger. Okay? It's too big to be efficiently gotten over by pushing and pulling. So what the cell does is it doesn't try to reverse the single reaction. Instead, it uses 2 separate reactions that are kind of a sidestep to get around that. Each of those steps requires a triphosphate. So it takes 2 triphosphates to get around that pyruvate kinase reaction of glycolysis. And the pyruvate kinase reaction only gave us 1 triphosphate. That was ATP. Okay? Well, let's look at these now. So this sets the stage for the very first reaction. The first reaction of this process does not occur in the cytoplasm. The first reaction of gluconeogenesis actually takes place in the endoplasmic reticulum. What am I talking about? Not endoplasmic reticulum. It takes place in the mitochondria. I have a cold, so my cold medication makes me even loopier than I usually am today, folks. Okay, the first reaction occurs in the mitochondria. All right? Mitochondria. Now, this reaction is catalyzed by the enzyme Pyruvate Carboxylase. That's what you see right here. And it's an interesting reaction, okay? You see we start with pyruvate that has 3 carbons. We add a carbon dioxide, which means this guy over here has 4 carbons. Okay. It takes energy, ATP, all right? And we make this guy over here called oxaloacetate. Oxaloacetate is kind of like pyruvate. It appears in many pathways. We'll see it occurring in a couple of pathways later this term, all right? But oxaloacetate has 4 carbons. It's an intermediate we did not see in glycolysis. So the first step of the 2-step process to get around pyruvate kinase reaction, going upwards, is to make oxaloacetate. Now, the other thing that's interesting here is this reaction requires biotin. Biotin is a coenzyme. Biotin is a vitamin. Many coenzymes we talked about before are vitamins. And if you recall, which you probably don't, but if you recall, this was an example of a coenzyme I talked about earlier in the term where I gave it a function. And I said whenever you see a biotin in a reaction, it almost always is involved in grabbing a carbon dioxide and helping it get attached to something. And that's what's happening here. It's actually getting attached down here. All right? Carbon dioxide is being grabbed by biotin and being attached onto the end of pyruvate to make oxaloacetate. Another clue that this is a biotin-containing enzyme is anytime you see the word carboxylase in an enzyme name, that enzyme is putting a carboxyl group onto something. That is, it's grabbing a CO2. It's almost always the same thing. It's going to be needing biotin. Okay? Hi. Oh, you're looking at the stuff. Yeah, thanks. Don't know what's going on with it. Okay. So, let's see. Where was I? So this shows the process that's occurring. Magic.
The shutters were closed for some reason.
Well, 2 down and... Thank you, Obi-Wan Kenobi. All right. Thank you much. All right. So now we have our 3 screens again. All right. So this shows a little bit of what I've just been describing to you happens. There's pyruvate that's out in the cytoplasm. Remember, all of glycolysis occurs in the cytoplasm. Pyruvate has to be moved into the mitochondrion. And we'll see later that's also important if we decide to oxidize pyruvate instead of making it into glucose. If we oxidize pyruvate, it occurs also in the mitochondrion. So that's a very convenient move. Pyruvate's in here gets converted to oxaloacetate. That's the pyruvate carboxylase reaction. Okay? You don't need to worry about all this other garbage here. Ultimately, oxaloacetate makes it back out. And the rest of the process, with the exception of the very last step, occurs also in the cytoplasm. Okay. Here's biotin. And no, you're not going to memorize the structure of that, but that's what it looks like. Biotin gets attached to a lysine. That's what it's showing you right there on the enzyme. It's a covalent attachment. Now, I said we did a 2-step. You've seen the first step of the process. Now you're going to see the second step of the process. We're aiming to get back into the glycolysis pathway going backwards, which means we're trying to make PEP. We're trying to get back from pyruvate to PEP. And now with this second step, we see that this is exactly what happens. Here's oxaloacetate. Notice that we have another triphosphate. In this case, it's GTP that's necessary for energy. That's why I said we had to use triphosphates, and I didn't say ATP. We're using GTP here to do this. The product of this reaction is phosphoenolpyruvate, which is what you saw before, PEP. The carbon dioxide that went on comes off, and we're left with GDP. Now this enzyme, Phosphoenolpyruvate Carboxykinase, you can call PEPCK. PEPCK. But if you spell it PEPSIK, we'll count it wrong. So make sure you get the acronym right? PEPCK. This enzyme is found in the cytoplasm, and it's not found in all cells. This is an important thing. Okay? It's not found in all cells. In fact, it's primarily found in the liver and in the kidney, some cells of the kidney. Okay? What does that mean? It means that most cells of our body cannot do gluconeogenesis. Most cells of our body cannot do gluconeogenesis. If one enzyme's missing, we're hosed. Okay? So it means that our liver, which does do gluconeogenesis, is very important for helping our body to maintain those blood glucose levels, because that's one of the two mechanisms we have of making glucose there. Okay. Well, what we've done so far is right down here, pyruvate to PEP. All right? We've gone from here, one step, there, two step. It's taken us two triphosphates. It's taken us an ATP here, it's taken us a GTP here, and we're back at PEP. Now I want you to remember that in order to make glucose, we're going to have to start with two pyruvates. So we've got to double everything going up as well. So if we're going to have two pyruvates and two PEPs, we're going to need two ATPs and two GTPs going up. Okay? This also shows us and reminds us that when we talk about a pathway being a man-made definition, here's lactate, right? Lactate can exchange with, if we have lactate, we can easily convert it into pyruvate by reversing that reaction that we saw before where we reduced pyruvate. Now if we oxidize, excuse me, if we oxidize lactate, we make pyruvate. Okay. You also notice over here that amino acids can feed into here. Amino acids. I've already told you that alanine is readily made from pyruvate. Okay? Pyruvate can readily be made from alanine as well. So we break down protein, we have plenty of alanine. It'll go over here and can get made into glucose. That's one of the reasons why a person on a high protein diet, all right, has plenty of glucose, because they can convert amino acids into intermediates here. You see amino acids go over here to oxaloacetate. Aspartic acid is readily converted into oxaloacetate. And vice versa. Oxaloacetate is readily converted into aspartate. Okay, so we're now back at PEP, and now we're going to start reversing the process. How do we reverse the process? Well, this is really easy. We don't have to do anything. All we have to do is make plenty of this stuff, and what happens is the concentrations reverse, 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 and we just climb the ladder. We use the same enzymes that we used in glycolysis for all of these other reactions. Okay? Same enzyme that we use for glycolysis in those reactions. We're just moving up the ladder is all that we're doing. Okay? Make sense? Okay. Now, that means we also have to reverse the ATP going back to ADP, right? So now we're going to burn a couple more ATPs going up this ladder, right? So far we've burned 4 triphosphates down here for those 2 pyruvates. And now these 2 pyruvates are each going to take an ATP going up, which means there's 2 more triphosphates. We're at the count of 6 triphosphates so far to make glucose. It takes more triphosphates to make glucose than we get out of glucose when we burn it. Okay? That's what we have to eat. If we relied solely on that going around and around in a circle, we'd soon run out of triphosphates. All right. Well, we get up here, and we've got 2 glyceraldehyde 3-phosphates. One of them gets converted into DHAP. And now these 2 guys come together and make fructose 1,6-bisphosphate. And in this direction going up, the aldolase reaction is very favorable. Whenever we see something that has a large delta G 0' positive in one direction, In the other direction, it's negative the same value. So aldolase is very favorable, makes plenty of fructose 1,6-bisphosphate. Well, it's at fructose 1,6-bisphosphate that the process needs a different enzyme. It needs a different reaction. Okay? I'm going to tell you why that's the case. All right? If we were to simply reverse the process of glycolysis, what would we be doing? Well, we would have to reverse this process and convert ADP back into ATP. Right? Because remember, in this going down, we were making ATP. So we're using ATP. If we're going to go backwards, we've got to make ATP. Well, this doesn't make any sense. Why doesn't it make sense? Well, we're making glucose. When will our cells be making glucose? When they have high energy or they have low energy? No, they're making glucose when they've got plenty of energy, because it's taking energy to make glucose. The rest of the body might need it, but the liver cells have plenty of energy. They're going to be making glucose for the rest of the body. They've got high energy. Their ATP levels are high. They've got plenty of triphosphates for down here. They've got plenty of triphosphates down here. They're loaded with triphosphates. Do they want to add to the triphosphate pool? No, they don't. So rather than try to fight an energy barrier here, they say, we're going to do a different reaction. And that's what they do. So they have a different enzyme and a different reaction. So instead of making ATP, all they do is they clip one of the phosphates off. They just clip it off. And that clipping the phosphate off turns out to be energetically favorable, because remember, these two phosphates are negative, right? It's energetically favorable. We get rid of one of the phosphates. Oh, goodbye, get out of here. This enzyme is one of the four enzymes that's different. Okay? Instead of having PEPCK, which is the enzyme that catalyzes the glycolysis reaction, this enzyme is called fructose 1,6-bisphosphatase. You can call it F16-BPase. Now notice that this reaction, as I said, is not generating ATP. The liver cell has plenty of ATP. It doesn't want any more. There's no reason to continue to do this. Okay, now I'll stop and take questions before I move on.
What's the abbreviation for that enzyme?
The abbreviation for the enzyme, F as in Frank, 16BP as in the gas company, ASE, ASE. F16BPase. Stands for fructose 1,6-bisphosphatase. Question?
Was biotin only in the first step?
I'm sorry?
Was biotin only in the first step?
Was biotin only in the first step? That's correct. The only place that this process uses biotin is in that very first step. Yes?
So I really can't see that diagram at all, and I don't have a little one in front of me.
Okay.
It flips a phosphate from
Yep.
fructose 1,6-bisphosphate
Yep.
to make fructose 6-phosphate.
That's correct. So it's clipping the phosphate off of position number 1. Right. So it's the exact, well, I shouldn't say it's exact, but it's the reversal of glycolysis, except for it's not making ATP. So it is a different reaction, and this different reaction is energetically favorable because those phosphates went out of there. Okay. Yeah. Yeah.
So, you said something about how it takes more triphosphate to make glucose
Yep.
than...
Then you get out of it.
Okay. I just wanted to...
That's right.
That's,
So let's think about that. It's a good question. Let me just elaborate on that just a minute, okay? It takes, if we add up all the triphosphates it takes to make glucose in gluconeogenesis, okay, it takes 6 triphosphates. If we look at all of the triphosphates we get out of the breakdown of glycolysis to pyruvate, we get 2 triphosphates, 2 ATPs. So let's think about this for a second. Let's imagine I've got a cell, and it's relying on gluconeogenesis and glycolysis for its only source of energy. It starts with glucose. It breaks down glucose, and it gets 2 ATPs. It says, okay, now let's go back and make glucose again. So it's got to run back up the gluconeogenesis, and it's got to burn 6 triphosphates. Right? It's in the whole 4. It burns and gets 2 more, and then it's out 6 again. Every time it does this whole circle of glycolysis, followed by gluconeogenesis, it's losing 4 triphosphates. Okay? Not a good idea. Not a good career move for the cell. So if a cell were relying on this only for energy, it would burn itself out. It would have no energy. Cells have to have other source of energy, and that's why I said we eat. So our food is providing the excess source of energy that's making up that gap. If we don't eat, we lose weight. Duh, right? We know that. This cycle that I just described to you has a very important name. It's called a vicious cycle or a futile cycle. It's futile because every time it turns, nothing happens except for you lose 4 more triphosphates. That's really bad. So it turns out that cells have some pretty elaborate controls inside of them so that both are not going on at the same time. Because if both were going on at the same time, all that the cell would be doing would be losing triphosphates and making heat. That's all it would do. So cells have things set up so that they will only have one process going on at a time, either glycolysis or gluconeogenesis, but not both. Okay? And we'll see when we talk about regulation how and why that's the case. Yes?
That cycle doesn't actually happen in nature.
That cycle doesn't happen to any significant extent inside of cells. That's correct. Yep. It would be a futile cycle if it were. Let me also, since you asked the question, you guys have some good questions here. Let me just elaborate one more thing. Cells do occasionally use futile cycles. There's other futile cycles that I won't talk about here, but I'll mention them to you later. Other futile cycles that cells do, and they do them for a very important reason. Okay? So let's think about what's happening here. Okay? This cycle is going, and as I said, all it's doing is it's losing ATPs, or triphosphates, and it's generating heat. Okay? We have a type of fat in our body called brown fat that runs a type of a futile cycle we'll talk about later to generate heat. And brown fat turns out to be around our spinal cords. Keeping our spinal cords warm is important because the transmission of nerve signals is a function of temperature. If we start slowly, we're in trouble. Right? Thank you. I've worked hard on that. We can't slow those down. So even if we get cold and we're shivering, we still want to be able to respond to things that's out there. Otherwise, the polar bear comes and gets us, right? The polar bear has already got it all figured out, right? We're not adapted for that, so if our nerve signals are slowing down, we're not going to escape. Yes?
So to me, it's not really a futile cycle, it's a purpose.
It has a purpose in that case, that's correct. And the purpose is generating heat. But other than that purpose, that's about the only purpose it has. Yep. Okay. Oh, okay, so I haven't finished up here. Let's finish this last up here. So we get to fructose 6-phosphate. We reverse the glycolysis reaction, go from fructose 6-phosphate back to glucose 6-phosphate. And then we hit the last of the enzymes that are different. The last of the enzymes that are different is we do exactly what we did down here. Instead of trying to regenerate ATP, we simply clip off a phosphate. This enzyme is called glucose 6-phosphatase, or G6Pase. This enzyme is found, and this is the one that's found, in the endoplasmic reticulum. So the very first enzyme of the process was found in the mitochondrion. The very last enzyme of the process was found in the endoplasmic reticulum, and all the other enzymes are found in the cytoplasm. It's called glucose 6-phosphatase, or G6Pase. Yeah?
So carboxylase is also found in
PEPCK carboxykinase is found in the liver. That's correct. Yes. Okay. So that's the overall process. Now, I hope I've convinced you from the argument about the futile cycle that regulation of this pathway is very, very important. Okay? It's very important because if we don't regulate this pathway, we are in deep doo-doo. Okay? Now, there's several enzymes that get regulated in this pathway. One of them is called pyruvate carboxylase. Pyruvate carboxylase, pyruvate kinase we saw in glycolysis, right? And I told you that that enzyme was regulated, and we probably thought, why in the world is the last enzyme in the pathway regulated? What good does that do? It's a very important reason it's regulated. Okay? It's regulated because of gluconeogenesis. Let's imagine, if you will, I'm a liver cell, and I've just decided, okay, it's time to start making glucose because this guy's out running a marathon and he's going to need the glucose for his muscle cells. Okay? So we've got to start making some glucose for him. Let's start with pyruvate. And we go from pyruvate up to oxaloacetate, and oxaloacetate to PEP, right? That's the first two steps of gluconeogenesis. And if I get to PEP and pyruvate kinase is active, what's going to happen? PEP is going to go right back down to pyruvate. Right? So I go up 2 steps and I come down. I go up 2 steps and I come down. I've just made a little mini futile cycle. Right? I'll never get to make glucose if I do that. So cells have regulation of pyruvate kinase so that they don't have that enzyme active when it's climbing the ladder. When we're making glucose, when we're doing gluconeogenesis, we want pyruvate kinase to be turned off. Okay? We turn it off, now we don't have to worry about that little mini futile cycle. PEP then goes on up to 2-phosphoglycerate, and then we start climbing the ladder and making glucose. So regulation of that very energetic enzyme reaction allows the cell to do gluconeogenesis. Very important point. Everybody understand that? Now, pyruvate kinase is interesting for another reason. Okay? Now, I'm not going to hold you responsible for phosphorylation, dephosphorylation. I don't really care if you know that. That's not a big thing for our purposes. Okay? But something that is big for our purposes is this guy. Well, first of all, before I talk about this guy, all right, ATP, right? ATP is going to turn this enzyme off. We don't want to be breaking this down as we're making more, right? As we have plenty of ATP, we don't want to be breaking this down to make pyruvate. We want to be taking pyruvate and going up, right? In gluconeogenesis, we've got plenty of ATP. So we want this enzyme turned off, so we're not making pyruvate during this time, all right? This guy you've seen before. This guy was an intermediate in glycolysis. Now, this is really cool. Remember this guy. Let me show you glycolysis again. Fructose 1,6-bisphosphate was an intermediate in glycolysis. Here was the reaction in glycolysis. All right? Here it is right here, fructose 1,6-bisphosphate. Anybody remember the reaction or the enzyme that broke down fructose 1,6-bisphosphate? Aldolase, right? And this was the energetically unfavorable reaction, right? And this was the one that took the pushing and the pulling, right? I'm going to tell you how the pulling occurs. Okay? Here's how the pulling occurs. Well, let's imagine that I'm out sitting in my yard, or maybe I'm sitting in this room, and the building catches on fire. Okay? The building catches on fire. I've all of a sudden got to get the heck out of here. My body starts dumping glucose into the bloodstream so I can get out of here because the adrenaline is going to release a lot of glucose into my bloodstream. Right? My muscle cells say, okay, this guy's getting out of here. We're going to start breaking glucose down to glucose 6-phosphate, and glucose 6-phosphate down to fructose 6-phosphate, and fructose 6-phosphate down to fructose 1,6-bisphosphate. We can start increasing the amount of reactant. Right? There's the push. We see the push. The push is coming because the body has dumped glucose into our cells. We start pushing this, that is increasing the concentration of fructose 1,6-bisphosphate. But I told you to get over this hump, we had to not only push, we also had to pull. Now you're going to see how the pulling occurs. With only pushing, what happens? Fructose 1,6-bisphosphate levels start going up. And when they start going up, what happens? It comes down here and activates pyruvate kinase. Pyruvate kinase says, oh, you want me to convert PEP into pyruvate? Okay. And the PEP concentration starts to fall. And when it starts to fall, the concentration of 2-phosphoglycerate starts to fall, which means that the concentration of 3-phosphoglycerate starts to fall, which means the concentration of 1,3-bisphosphoglycerate starts to fall. You start to see where this is headed? Now it gets up to right here, and the concentration of glyceraldehyde 3-phosphate starts to fall, and that's pulling. Now this is a really unusual regulation. There aren't many places in cells where do this. This is an example of what I call feed-forward regulation. Feed-forward, that is, an intermediate in the pathway is activating an enzyme later in the pathway. So feed-forward regulation occurs when an intermediate in the pathway activates an enzyme later in the pathway. This is how pulling actually occurs. So through the combination of pushing and pulling, that aldolase reaction is overcome. Make sense? Yeah, Lisa.
Which enzyme did you say fructose 1,6-bisphosphate activates?
So fructose 1,6-bisphosphate activates pyruvate kinase, the very last enzyme in the pathway. Yes?
What did you say was called when an intermediate product activates reaction to the known pathway?
Okay, so when an intermediate in the pathway activates an enzyme later in the pathway, it's called feed-forward activation. Because then I know that feeling. I've been hacking and coughing myself the past 2 days. I think I got it from you, actually. I got it from somebody in here. What's that? Okay. All right. So this is cool. All right. Now, I want to show you something that's even cooler and easier to understand. Any questions on this before I go forward? Did anybody in this room go and see the play at the University Theater earlier this year about Gertie Corey? Anybody? One back there. Okay. This will make sense to you. So Gertie Corey was a very famous woman scientist. She and her husband worked out some very interesting things about some of these pathways that we're talking about right here. And she and her husband discovered a cycle that was named for them. And Gertie Corey is a very interesting woman. She had to fight very hard to be recognized as a female scientist because in the times of the 40s when this was going on, women were not respected as scientists. Well, the Quarry Cycle... Oh, actually, before I do that, I'm sorry. I'm forgetting one other thing. This guy right here. All right. This guy looks like something that you've seen before, but it's not. It's not fructose 1,6-bisphosphate. It's fructose 2,6-bisphosphate. Why do I show you this? This turns out to be one of the most important regulators of glycolysis. This molecule, okay, activates PEPCK and it inactivates fructose 1,6-bisphosphatase. And it does this in very, very tiny concentrations. Okay, so yeah, I'll show you in just a second. So the molecule is fructose 2,6-bisphosphate, or F26P. Let's look at where it works on the pathway.
Should it be BP or just P?
It should be BP. Did it say P?
Yeah.
Oh, it's BP. Yeah, sorry. Okay. It activates PEPCK. It inhibits fructose 1,6-bisphosphatase. It's having opposite effects on the two enzymes that are involved in glycolysis or gluconeogenesis. Right? This is a prime example of what we call reciprocal regulation. Reciprocal regulation. Reciprocal regulation occurs when one molecule has opposite effects on catabolic and anabolic pathways. I'll repeat that. Okay? Reciprocal regulation occurs when one molecule has opposite effects on anabolic and catabolic pathways. What's PFK going to do? It's going to turn on glycolysis. It's going to turn off gluconeogenesis. It's beautiful. We don't want them both going on at the same time. One molecule takes care of both of them. Bang. Now this molecule, as I said, is one of the most important regulators because it's used in such tiny quantities, okay, that it has enormous effects on enzymes, even in very tiny quantities. Okay. Okay. Questions? Reciprocal regulation. Yeah. Fructose 1,6-bisphosphatase right there. The two enzymes on either side of this thing right here. Okay. Now, I want to tell you about the Quarry Cycle. You'll like the Quarry Cycle. You'll understand it very readily, okay? This is fun. You understand how your body works, okay? Let's imagine I'm out running a race. I was out running a race. I ran the Mad Scientist 10K this past weekend, and believe me, my Quarry Cycle was going. Okay, I was hurting about halfway through that 10K run. My muscle cells were not getting oxygen as fast as they were needing it. So what happened inside of my muscle cells? They had to keep contracting because I wasn't about to stop, being the stubborn cuss that I am. I'm not about to stop. They've got to keep contracting, so they have to make, what molecule do they have to make? If they don't have enough oxygen, what do they have to do to keep glycolysis going? What molecule do they need? NAD, guys. Remember? They make lactate so they can make NAD, right? They make NAD, okay? So they can keep glycolysis going. What are they producing when they're making NAD? They're producing lactate. And lactate is of no use to my muscle cells. They can't do anything with it. If they let it sit around and accumulate inside of them, they make lactic acid, they get all acidified, and they die. Well, muscle cells aren't stupid. Okay? They kick lactic acid out, and when they kick it out of the cell, where does lactic acid go? It goes into the bloodstream. And when it goes into the bloodstream, it travels to the liver. And it gets to the liver, and the liver says, oh boy, lactic acid. Liver being liver is one of your most perverse organs, by the way. Okay? It's a very perverse organ. It takes up lactic acid. Your liver is very different from your muscle cells. Your muscle cells are out here a long ways away from the heart. The oxygen supply in the muscle cells is low. The oxygen supply in your liver is high. The liver has plenty of NAD, and it says, oh, give me your lactic acid. I'll convert it back into pyruvate, because that takes NAD. And when I get to pyruvate, oh, well, I've got plenty of ATP because I've got plenty of oxygen. Let's make glucose. So the liver takes this lactic acid that the muscle cells are dumping out into them, makes pyruvate, makes glucose, and then the liver cell says, I don't need all this glucose. It's a poison. Out into the bloodstream. And where does the glucose go? To the muscle cells. It's beautiful. Your organs are cooperating with you. This is called the Quarry Cycle, and that's what it's shown right here, okay? Here's what's happening, okay? We have muscle cells that are breaking this down. Glucose going to lactate. Blood supply takes it up to here. Lactate in the liver gets converted to pyruvate, goes to glucose, and then gets dumped back in the bloodstream and back down here. That's what's keeping you going when you're running out of oxygen inside of your muscle cells. Pretty cool stuff. Questions?
How long does the Quarry Cycle last? Only a couple seconds, right?
Quarry Cycle lasts as long as you're lasting. As long as you're running out there, that Quarry Cycle's going on. Yep. You betcha.
Well, when you're sprinting, though, eventually you get to a point where
Yeah.
it feels like your muscles can't do anything.
That's right. So the question is, so sprinting is a very good example. The sprinting, when you're sprinting, there's actually something else that kicks into play. But let's use the Quarry Cycle as an example, okay? Even with this mechanism going on, the liver can't supply muscle as fast as the muscles would like to use it, okay? And so you can't maintain that sprint forever, and that's the reason you can't maintain that sprint, okay? We'll talk later about the citric acid cycle. When we talk about the citric acid cycle, we'll discover the value of oxygen, okay? The citric acid cycle, as I said, requires oxygen. When we have the citric acid cycle going, if we start with glucose and we make pyruvate and then we go through the citric acid cycle, that one glucose gives us 38 ATPs. And if we go through the Quarry cycle, glucose to pyruvate to lactate, that muscle cell is getting 2 ATPs. It's got to burn 19 times as much glucose to get the same amount of ATPs as if it were when oxygen was present. So here's a question I ask, and everybody gets it wrong. If you want to lose weight, you want to go aerobic or anaerobic? You want to go anaerobic, folks. And most people think of it as going aerobic because you start breathing heavily. But you're going anaerobic. That's why you're breathing heavily. If you think about it, you want to burn off excess stuff, right? You want to burn off all this weight. Don't you want to make ATP in the least efficient way? It's going to take more stuff to burn off. So when next time you hear somebody saying that they're going aerobic, correct them. A, they're going anaerobic, and B, if they want to lose weight, that's good. Now, I don't recommend they go put their head in the paper bag and breathe only nitrogen because that would not be a good career move. However, going anaerobic is really good for losing weight, and that's what you're doing when you're exercising. You're not going aerobic.
You can't maintain the exercise as long, though. So you could exercise at a higher rate for a longer period of time and burn more fat than going anaerobic right away, burning some fat and being totally wiped out.
I'm not sure what you just said, but the point is endurance is another factor here. Yes, okay. But good exercise will be long endured exercise, not sprints. Sprints generally won't be a good plan. Excuse me, a good plan for losing weight. But jogging a longer period of time will. I was going to do a song. I'll save it till next time. How's that? See you guys tomorrow. Oh yes, and the material for the exam will stop right here. What's that?
You see, like those, I have, like, when I run,